Sharing. Okay. All right. Hello, everyone. Thank you for coming. I know it's a little earlier than usual, but glad you could all make it out. My name is Clarence Mah. I'm a PhD candidate at UC San Diego in the labs of Hannah Carter and Jean Yeo. And today I'll be telling you a bit about my work in developing methods, computational methods, for decoding subcellular RNA organization from spatial transcriptomics data. A brief outline before we get into this. I'll give you a little bit of background on some recent systematic approaches to characterizing RNA localization. And then I'll touch on my work on methods. And then I'll show a use case where we've applied this to study a particular system. So from transcription all the way to translation, RNA is regulated both spatially and temporally. Every stage of the RNA life cycle is linked to a different place in the cell with its own machinery, from transcription, which is affected by chromatin accessibility, as well as organization, all the way to splicing, in which transcripts are converted from nascent mRNA to mature mRNA, which are then packaged into these ribonuclear protein granules in the nucleus before they're exported onto different parts of the cell, such as the ER, where they're translated by ribosomes, or into the cytosol. And from there, they undergo both passive and active transport to be translated or transported to specific areas of the cell. And finally, they're degraded after they've produced the proteins. So as you can see, RNA is heavily regulated by both the space and point in time in which they exist in the RNA life cycle. A recent study that was able to characterize RNA localization at the transcriptome level was performed by Alice Ting's group, in which they developed a new technique called APEX-seq, where they have been able to proximity label RNAs to several subcellular compartments or membranes. And by biotinylating these transcripts, they're able to pull them down and sequence each fraction and look for enrichment at these various compartments. And from here, they've pulled out more than 3,000 differentially localized genes to one or more of these compartments, and have given us a little picture of what we might see at the subcellular level, as opposed to at the cell-to-cell variation level. Similarly, in this study by Smolik et al., they developed a new technique based on time-lapse-seq, which is essentially a metabolic labeling technique that is able to measure RNA half-life across the life cycle of RNA. They've basically improved this to work at the subcellular level by combining it with fractionation techniques, pulling out both subcellular fractions as well as RNA half-lives at the compartment level. And from there, they're able to extract the RNA, convert it to convert the 4SU labeled transcripts to Cs, and perform short-read sequencing. And from these quantifications, they were able to model what they call RNA flow, or dynamics of RNA across the life cycle. On the other side of things, in the imaging world, there have been a number of developments, starting with fish-based techniques. In this particular study, they did a high-throughput single-molecule fish experiments to identify locally translated mRNAs. And you can see in the top, there are a couple examples of different localization patterns of RNA, which they have found to be associated with translation as well at their points of localization. So bringing it back to RNA sequencing, in the development of these high throughput technologies from RNA-seq to single-cell RNA-seq to now spatial transcriptomics, we can now get a better and better, more granular measurement of RNA, now down to the subcellular level. And so my project is asking, can we push these existing spatial technologies further and really use it to study cell biology? To study that, we need appropriate spatial analysis methods to fully take advantage of this data. And that brings me to my work, Bento. Bento is a toolkit I've been developing to ingest all kinds of RNA spatial data, from multiplex fish imaging based to slide based, as long as there's molecularly resolved and subcellular resolution, and puts all this in a standardized data format and is able to then apply our machine learning and statistical analyses to study the cells. And today I'll be focusing on part of this in which we characterize molecular gradients in subcellular compartments. This technique in the toolkit is called RNA flux, and we found that this was able to quantify these subcellular transcriptional gradients. So here we have a picture of a cell where the boundaries of the cell membrane and the nucleus are delineated in white, and each dot is a single molecule, and the color denotes what gene species it comes from. It turns out if we look at the local transcriptome or expression at a given point in space, we can use that to characterize how it changes across the area of the cell. So following some normalizations, we can convert this embedding at every point in the cell to a lower dimensional space for interpretability via PCA, and further clustering finds these domains. So the red, green, and blue denote the first three principal components of this embedding. And you can see they kind of correspond to different areas of the cell. And more convincingly, if we apply this method not to one cell, but multiple cells, we can see that we get these consistent embeddings across all of the cells at the same spatial locations. For example, red kind of highlights the nucleus, and this yellow, which is a mix of two colors, kind of highlights the periphery. And then we can apply the clustering techniques, in this case, a self-organizing map to cluster in the high-dimensional space and identify these domains. Here, the colors denote separate domains, and you can see that the separate domains are present consistently, again, in all the cells. So we want to characterize these domains. How do we do that? Bringing back the APEX-seq data sets in which they found RNAs enriched in various subcellular compartments. We can then take those enrichments scores of these 3,000 or so genes and overlap that with our panel of measured genes in our spatial transcriptomics experiment. In this case, we've measured 130 genes, in which 60 overlap with these 3,000 in U2OS cells. So as you can see, when we compute these enrichment scores, which is simply a weighted sum of the subcellular enrichment with our embeddings, we see highlighted regions of the cell that correspond to our inferred domains. In this case, we're visualizing the outer mitochondrial matrix in correspondence with one of these flux maps, as we call them, flux map 1. And if we generalize this across all our inferred domains and the 6 or 7 various gene sets, we can get a score for every compartment in every domain. And here I'm highlighting the nuclear gene set, which again corresponds strongly to FlexMap2. And here's another example of a slightly weaker signal. Doesn't correspond as well, but we can find the enrichment of the ER corresponding to FlexMap3, the yellow one here. And FlexMap 4 and 5 are cytosolically enriched. So now we wanted to apply this to study cardiomyocytes, specifically doxorubicin-induced stress in cardiomyocytes. So very briefly, doxorubicin is a cancer chemotherapeutic, and it is extremely cardiotoxic when given as a treatment, meaning it has very strong side effects. And it's very important because it's actually given to 60% of pediatric cancer patients, as well as 35% of breast cancer patients. But the cardiotoxic side effects result in 7 to 10% of childhood cancer survivors developing heart failure within 30 years, and a pretty high mortality rate for patients who develop cardiac failure. But the mechanism for how this happens is still not completely known. So it's known, though, that doxorubicin perturbs many of the cardiomyocyte pathways, but we're not sure what mechanisms. And so our hypothesis here is that RNA localization is in fact affected and hasn't really been measured before. So we had a simple experiment where we had a vehicle treatment in which treatment was treated with doxorubicin for 12 hours in iPSC-derived cardiomyocytes, and we performed a spatial transcriptomics experiment with Resolve Biosciences with their technology, molecular cartography, very similar to MERFISH. And we targeted 100 genes with cardiomyocyte-related functions, apoptosis, calcium signaling, so all very related genes. So first thing we did was we had to segment the data sets to get cell boundaries and nuclear boundaries. And we did that with some pre-existing tools, CellPose and ClusterMap. And we found roughly, able to recover roughly 20,000 cells across both conditions and 2 replicates. And we found some differentially expressed genes, but this we expected, but we wanted to see what more we could find. So we applied our RNA flex method to this data set, and a little hard to see, but the nuclei strongly correlate with the same color across all these cells, which was promising and showed that we are capturing intracellular variation. And if we went further to characterize each domain that we pulled out of our embedding, we could again apply the same organelle compartmental gene sets to try and annotate these domains. So for example, the red here kind of corresponds to the nucleus. So yeah, we can do the same thing for the treatment data. As you can see, it's relatively similar to our vehicle. And finally, we can compare how do transcripts move between domains before and after treatment. And in fact, we found a couple important genes for cardiomyocyte function, including RBM20 and CACNB2, important for splicing, specifically in cardiomyocytes, and calcium signaling, as well as a couple of cell growth related genes. And what we found was that these genes moved from what we think is the ER, from looking at enrichment of our domains, to remain in the nucleus, which is very unexpected. And so RBM20 is already known to perturbs splicing when it's dysregulated and leads to heart failure. So we think this is a very promising candidate for some follow-up studies, and we're currently working to understand what this nuclear retention of the RBM20 transcript is doing. So overall, this is kind of shows an application of what you might do with subcellular resolution data. And we've shown that doxorubicin leads to perturbation of RNA localization and of RBM20 and many other critical genes. And we're currently validating that. And very briefly, we're applying this work to more and more data sets. Here's a xenium data set of breast cancer tissue. So just zooming in on this, you can see that there is a density of transcripts, and we can start to apply these subcellular quantifications across the cells. And again, so we're improving our toolkits and kind of converting our data set to be compatible with SCVerse. We're a community member, and we hope to use spatial data going forward. And again, yeah, I'd like to thank everyone from my labs, Jin Yo's lab and Hannah Carter's lab, as well as the Center of Epigenomics for collaborating. Thank you.
Okay, this is very, very cool stuff. So two things. One is your initial maps. They look very nice, but also very crude.
Yes.
So is this reflecting, I'm just thinking this is reflecting the limitation mostly of the technology of mapping areas, and related to that, right, you basically do this sort of unsupervised, and then you go back with AIC and you say these are the known markers that are going here and there. So wouldn't it have been better to start with, rather than say sort of neighboring, right? Saying these guys are going where these guys are known, and that would have been a simpler, maybe more accurate labeling than doing the PCA and the maps and then going back to try and find out and report it in terms of trying to figure out new ones. And okay, so that's the first question. The second one is those changes that you showed in the second part of the talk. Could that be related to changes in isoforms? Or, and the related one is, are you mapping differences in isoforms in this?
Yeah, yeah, those are good questions. So I'll answer the second one first. So in that study, we measured 100 genes, not at the isoform level, so it's not clear if those changes are isoform specific, but that's definitely something we want to follow up on.
We know splice factors specific ones have isoforms, localized different.
Yeah. Yes, yeah, exactly. Though, yeah, that gene itself is the splicing factor, so it will be interesting to see what the downstream effects of that are. And I guess back to your first question, yeah, so I think the issue with looking at those genes directly are because we wanted this to be a generalizable method, and not every single spatial experiment is going to measure the same genes. Number of genes or even gene panel could be completely disparate. And the advantage of using this enrichment method is because there are 3,000 quantitative measurements of localization. So we can look at relative rank, relative enrichment of genes that may or may not even be in the gene set that correspond to those compartments. But yeah, I'm happy to talk more. Thank you.
Thanks. So cells are not perfectly flat, which means that when you have the spatial transcriptomic data, you will have noise. How do you handle that?
Yeah, great question. So the data sets I showed today are all from cell lines in a dish. So for the most part, they are pretty flat in a monolayer. The cardiomyocyte data set I showed, we actually did have some issues with the later time points being very dense. So cells kind of sitting on top of each other. So I didn't show that data today, but yeah, it's definitely a valid concern and kind of shows the need for 3D based spatial measurements. And that's going to be a big issue for tissues going forward. Yeah, thanks.